Alrighty. Well, you thought maybe since I was gone for the track meet, you might get out some work. Well, tough luck, not today. So I made this awesome video for you here so we can continue to talk about polymerase chain reaction. So hopefully this video helps you, and there's some other things that you need to do after the video, what you can do in your spare time. So check it out at the end, and hopefully enjoy it, and hopefully you learn something from it, and hopefully the video works out. But once again, to continue on with polymerase chain reaction. Yesterday we talked about the introduction of PCR. It does stand for polymerase chain reaction, which is an in vitro technique used for amplifying DNA, a very specific part of DNA. In vitro, once again, means in a test tube. All right. Before PCR, DNA of interest could only be amplified by overexpression in cells, and this was limited yield. So the PCR process overall, once again, helps us amplify very specific regions of the DNA, so we can get clean, concise results. Let's see, here we go here. All right. Once again, we talked about the history of PCR and these individuals and their contributions towards PCR. Once again, the most notable thing is Thermus aquaticus and how we were able to harvest the Taq polymerase from Thermus aquaticus, which is that bacteria found in the Yellowstone National Park. The reason why it was so important is because we needed to heat up the DNA, but the only problem was heating up at such high temperatures caused most of the polymerases and other enzymes to denature. Thermus aquaticus, being it was in a hot environment already, could withstand the temperatures, especially its polymerase. So they were able to synthesize that in a lab and use now once again worldwide. So we're going to start talking about the reaction components. There are a couple here. The first is DNA template, the primers, enzymes, DNTPs, magnesium, and the buffers. So you might be asking yourself, what are the DNTPs? Those are basically spare nucleotides. So they could be adenines, guanines, cytosines, whatever they need to be. They're just extra ones. So DNA nucleotides is basically what it means. Okay. First off, we have the DNA template. So the DNA containing region to be sequenced. So your desired gene of interest, whether it be a cytochrome C gene, or if it was a gene for eye color or height, whatever it may be, we have a target sequence we're going after. All right. The size of the target sequence DNA can be amplified up to 3 kilobase pairs, which means about 3,000 base pairs. So that's quite a bit. If you get certain polymerases and stuff and certain reactions, you can get even more, which you're going to find out when you start looking at GenBank. But about 3,000 is pretty standard or basic. So step 2, you have the primers, and you have 2 sets of these primers. All right, they're generally about 20 to 30 nucleotides long, and they are synthetically produced in a lab. All right, and they're complementary to the 3' ends of the target DNA. They're not complementary to each other. So the reason why you want them to be about 20 to 30 nucleotides long, if they're too short, they could end up binding anywhere on the DNA, which is not good. So you want to kind of make them specific, but not too specific. Otherwise, there could be variations within, like mutations, that would render them useless. So 20 to 30 is about what you want. And you might be thinking, where can we find these or make these synthetically in the lab? Well, if I go back here, there are actual websites out there that can actually build custom primers for you. So you start researching a gene, maybe somebody has already looked it up, and you can actually basically email these people what you want, and they will synthetically produce it in a test tube and fill it full of these primers and then ship it back to you, and then you can begin your process. Now, we don't have to worry about that for our lab because the BioRed company has already made the primers for us, so all we have to do is add them, and then it'll start going. Okay, so going back here. So now we're back on track. So, and you do not want them to be complementary to each other as well. And the reason for this is because if they're complementary, they're going to stick together, and you're going to have these things called primer dimers. and that will ultimately just cause all the primers to stick together and not to your DNA, which renders it pretty much useless at that point. So you waste a lot of time and money to do something that just didn't work out well. So, as you can see here in the picture, you've got the template strand of DNA, and you've got the region to be copied. And notice there are a few extra nucleotides on the end that you don't necessarily need. So you get your first primer, and these are called forward and reverse primers, because one will go one direction, one will go the other direction. They'll actually anneal to the DNA, and they will copy that target sequence that you're looking for. All right? So on both sides of the DNA. All right, so now, step 3, enzymes. Usually Taq polymerase or any one of the natural or recombinant thermostable polymerases are used in this process. The reason why we use Taq polymerase, once again, is because we have to heat it up to about 95 degrees Celsius, which is about 203 degrees Fahrenheit, so it's pretty warm. So Taq polymerase is stable up to those temperatures, and therefore we can use it safely in the lab and get our target sequence DNA. It's very important for PCR process, and the reason once again we use it is because most enzymes or proteins would denature at this temperature. So this Taq polymerase has once again changed the face of how we study genetics. The PCR cycle is comprised of 3 different steps. The first step is denaturation of the DNA at 95 degrees Celsius. So once again about 200 degrees Fahrenheit. Number 2, the primer hybridization, annealing at 40 to 50 degrees Celsius, which is a lot lower than 95, so it's cooling it down. Number 3, DNA synthesis, the primer extension at 72 degrees Celsius. So essentially what we're doing is we're heating it up so that the DNA can break apart, so break those hydrogen bonds apart. Then we're cooling it down so that the primers will actually stick to the DNA, the strands now. And then we're going to heat it back up a little bit so that Taq polymerase can start its job. Because at the lower temperatures, Taq's not going to start synthesizing new strands. It's just going to be kind of dormant. Once we heat it back up, it's ready to go. So you might be asking yourself, what does a PCR machine actually look like? Well, there's not much to it. It's just a machine where it has a lid, you open it up, and you have these little plastic tubes that are made for PCR, and you stick them right in there. Once you close it, it's got a little heater on the top and within the bottom, so it can heat the samples up rather quickly, and it can cool them down rather quickly. So not a whole lot to the machine. It looks something very similar to this. Ours is a little less fancy and has a lot less little spots for the little test tubes. Now, so let's talk about this whole process here. So in the PCR cycle, you have step 1, which is denaturation, the template DNA by heat. Okay, at 95 degrees Celsius, again, about 200 degrees Fahrenheit. So we have our target sequence of the DNA, and we want to break those little hydrogen bonds right here. These little guys are right here. I want to go right down the middle and cut those things up. All right? So as we do that, that'll cause the DNA to break apart, and then therefore we can get our primers in there and they'll attach to it. So if you look at step 2, the temperature is lowered and the primers anneal to target sequences. So as you can see in the picture, if we look, we have our target sequence right here, and we want to attach our primer, which is right here, to it. So once again, they're 20, 30 nucleotides long. It's going to anneal right to it. You also have your second primer over here, which is amplifying the other complementary strand of the DNA. Once again, they're reading the DNA from the 3' to the 5', going in both directions at the same time. Now you might be asking yourself, what is the Tm up at the top? So temperature is lowered to Tm, which is basically the melting point. So basically we want to take the DNA, rip it apart with that 95 degrees Celsius, then drop it down enough so that those primers will actually stick to it. So you got about half your primers on there ready to go, which is good because without the primer sticking, we wouldn't be able to amplify it. Alright, so now you've got your next step here, which is to, at 72 degrees Celsius, Taq polymerase catalyzes primer extension as complementary nucleotides are incorporated. So as you can see, once again, we have the primer attached, fully attached, and our Taq polymerase right here is going to start synthesizing it in this direction and also in this direction. Once again, Taq polymerase can add about 1,000 nucleotides in about 10 seconds. So it works pretty fast. And keep in mind, there's not just one little Taq polymerase in a test tube. There's a whole bunch of them working at the same time, trying to do this job of synthesizing a new DNA strand for us. All right? So the results in 2 copies of the sequence. So we had our target sequence, and now we have a complementary strand. This is basically like DNA replication in a test tube. All right? But we're not done, because we don't want just 2 copies. We want a bunch. So target amplification, when we look at this, all right, we see at the top we start up there with one little bitty strand of DNA. As we go down, each cycle will create an exponential growth. So as you can see on the chart, we've got 2 here after 1 cycle, but look at what happens when we get down to 30. It grows so exponentially that we end up with, after 30 cycles, 1 billion copies of our target sequence. 1 billion. That's a lot. So when you start to amass that much DNA, you can actually put it in a little well in your gel and actually see it show up, which is pretty amazing. So PCR does allow us to do some pretty great things. Now, you might be thinking, well, how many times do we have to do this? Well, I'm going to back out here, and I can show you. This is from the actual lab manual that you guys will be doing or using for your lab. And this is what their standard procedure is for PCR. So you can see in the initial step, which is the denature, 94 degrees Celsius, and you do it for about 2 minutes, and you only do it for 1 cycle. Step 2, you get the thermal cycling, which is denature, anneal, and extend, which is 94, 52, 72. So very similar to what we just talked about. But when you do the denature, you do it for 30 seconds, right? And then you do the 52 degrees Celsius for 30 seconds, 72 degrees Celsius for 1 minute, and you do that 35 times. 35 times within that system, which is pretty amazing. All right, so you're getting, once again, over 1 billion copies of the DNA target sequence. The final extend is about 72 degrees Celsius for 10 minutes, and you'll do 1 of those. And then it says hold, which basically tries to keep the sample cold for quite a while so that you can come back and pick it up later. So if you need to go, get a cup of coffee, or, go to your next class, or, I don't know, take out the trash, what do you got to do, So anyways, back to this. So this is just a little graphical representation of the entire process. So you can see, as we go up to the top, you've got your step 1, which is the denature of the template. So notice the temperature is about 95 degrees Celsius. We drop it down quite considerably for step 2, the annealing, which you can see it's around about 60 degrees. Ours is actually a little lower, it's about 55 or so. But then it starts to go back up for the 3rd step, the primer extension. So we're starting to create our long strands. And then, once again, we start a whole cycle over again, starting with step 1. Then we go back down to step 2, then step 3. That's a terrible 3. 3, and then 1. Okay? So we just continue to do this for like 35 times. And that gives us our billion copies of our target sequence. All right. I'm sure there's a better way to erase this, but I haven't figured it out yet. Technology. So, let's see what else we got. All right, so you got the RT-PCR. So let's say you ran into this issue where you don't actually have the template strand of DNA, but you do have the RNA, like the messenger RNA. Could you actually get yourself a copy of the DNA, the actual gene? And the answer is yes. So this is called reverse transcriptase PCR. It uses RNA as the initial template. So the RNA-directed DNA polymerase, the RTH, yields of amplification products. Well, we are going to do one of these, which is the gel electrophoresis. So we can actually determine if we have DNA samples in there. And we're going to get into that process later on. Then we're going to do the sequencing of the amplified fragments. All right, well, you can, but we're not going to be able to do this because it costs a lot of money. But you can see in the picture down here, it is actually had the DNA sequence so that you can send it off to a lab, very similar to the one where you got the primers, and they can actually go forth, if you send them a sample, and then figure out what the D and TPs are, and then actually put it into a program based upon their amplified lines. So as you can tell at the top, each little nucleotide has its own color, and it creates a spike which the computer can read very quickly and then create your DNA sequence from that. And you can go through and you can use programs to download this information, and then you can upload it to GenBank, you can analyze it, you can compare it to other genes across the world that other scientists have looked at, whatever you want to do, which is pretty cool. There are also other methods, such as southern blot methods, to actually visualize and see the DNA fragments. So very similar to gel electrophoresis, but what you do is something just a little different. You actually take your DNA, step one, and you amplify it, do the same thing as PCR, and then you put it in your little gel, like so, like what we're going to do. But to actually see this, you actually put a piece of blot paper on it, okay, special type of paper on the top of the gel. It will then actually transpose the DNA fragments onto it, like so. And you can actually see them. And they're radiolabel probe incubated with membrane. You can see this. So they can actually tag certain things that you're looking for, and then you can actually see it right there on the film at the end. And you can analyze it and show it off and store it for long periods of time, rather than the gel that can only be stored for so long before the DNA actually starts to break down and dissolve out of it. Okay, so next, applications. Once again, the genome mapping and gene function determination, biodiversity studies, evolutionary studies on different genes or animals or species, whatever it may be. Diagnostics, parental testing of genetic diseases, early detection of cancer, viral infections. You can also do, once again, who's the baby daddy, stuff like that. Crime scene investigation shows, FBI forensics, determining who was the killer at a crime scene, and all that good stuff. You can do detection of drug-resistant genes, so you can find genes in, like, say, bacteria and determine how the genes have mutated and how they're becoming more resistant to antibiotics. All right, and then you can do what we're doing at the very end, the forensic DNA fingerprinting. So we're actually going to go and do a crime scene investigation so we can determine who is the actual killer based upon the information, the DNA given to us. All right, and yes, at the very end, you can take your DNA and you can compare it to other things, and you can build a cladogram like the one here, the picture shown here for different primates, which also includes us as humans. At the very end, you'll get to see some of that information. So advantages, though, it's automated. You don't have to do much to it. You just put the DNA and the primers and all that good stuff into the little test tubes, and it does it by itself. It's rather fast. It can take up to an hour and a half to actually amplify everything, but that's really pretty fast when you think about how much you're getting out of it. 1 billion copies of the DNA within an hour and a half. It's pretty awesome. It's pretty reliable and reproducible. So if you wanted to take a gene from a grasshopper and amplify it, somebody in Vermont could do the same thing and still get the same genes from that grasshopper using the same primers you used. Contained, less chances of contamination as long as you're practicing good sterilization techniques. High output, you get lots of it. It's very sensitive, which means it's pertaining to exactly what you want. There's many uses for it. Defined, easy to follow protocols. I mean, literally, you could show somebody how to do it that has never had any training on this or understanding of biology, and you could show them exactly how to do this, and they could do it, right? As long as they followed the protocols and everything to ensure the results. I mean, it's quite simple. A lot of medical companies are hiring people to do things like this. And they have very little training. And as they go through their program, they actually, some programs will allow them to get a degree in this area or a master's in this area, which is very cool. At the end here, all right, so at the end, I want you to get out your computers and I want you to watch the PCR video. So if none of what I made, or if nothing what I said makes sense, then at least maybe the video will help out and you can check it out there. All right. The next website I want you to check out is GenBank. We'll be using this later on, but you can see how people have used the process of PCR over the years to actually amplify all sorts of genes. And you can go through and type in anything that you want and make sure you spell it correctly, otherwise it won't come up, and see how many base pairs there are. It's huge, right? They're not just 1,000 base pairs, they can be up to 10,000 base pairs. Then we're actually going to be doing an activity with the phylogenetic tree thing there. Philo T, is a great little builder that you can. It actually uses GenBank and all that stuff on there to build cladograms. And if you want to play around with it, you sure can. If you're not sure how to use the Philo-T, there's another YouTube video below which you can check out in class as well. But you don't have to. It's just there for you. But it is pretty cool. So I encourage you to check it out. Hopefully the video was not too bad and everything went well. But ends up I probably won't use this because I'm sure the track meet got canceled. So yay, study time.